Should we start now? Welcome, everybody. Welcome to the session today on functional connectome. The organizer is Tingting Zhang from Pittsburgh, and I'm here to chair this session. Our first speaker is Mark Fiecas from Minnesota, and his work is on Changepoint analysis in a mixed model framework.
I'll be using the mic.
First, thanks to Hong Toon Jian for the invitation. I'm going to be talking about this Changepoint analysis, and by that I'll probably spend just a few minutes on it, primarily focusing on the questions that Jian posed in his email. One of which was actually, I appreciated the discussion in the previous session about when do we start with respect to these practices about improving our impact on the scientific community, on reproducibility, so on and so forth. And I'm actually going to discuss a little bit of that. And my answer, though, to that question is go earlier. I'll spend, I took Jian's questions and morphed it a little bit into my own. I'll spend a few minutes here on about training. And then I'll talk a little bit about change points, though this is going to be fairly non-technical when I get to that second point. So let's talk about training. I apologize, this is also a little bit of an advertisement. So 2 weeks ago, we finished an REU program called the Equitable Data Science and Adolescent Development. So this is an NSF-funded program, 10 weeks over the summer for 10 undergraduate students, where they worked on data from the Adolescent Brain Cognitive Development Study. So the ABCD study is the largest ongoing longitudinal study of childhood development in the United States, 11,878 people. They're going to be following them for about a decade or so. So we gave these 10 students access to that and have at it. To kind of give you, if you do work with undergrads and are interested more about this, we will be working on the applications. And we expect the deadline for that to be in January for next summer. So this is a program that I led. This was our very first time having REU. If you're not, especially for the biostatisticians here, if you're not familiar with an REU, you might be familiar with SIBS, Summer Institute in Biostatistics. It's pretty much that, but longer. And it's very project-driven. And more specifically, we had these 10 students. They worked in pairs. And here are the titles of their projects. Okay. So not all of these are brain imaging, but all of them are certainly brain imaging adjacent. The last two in particular are actually related to brain imaging. The other three are more along the lines of mental health. I don't know if I see Burned. I was talking to Burned earlier about normative modeling. That's this last one here, actually. They built a Shiny app. The code's available on GitHub. Everything is Quarto from the ground up. They learned everything from day one. This is how you produce reproducible workflows and documents, and that way anyone and everyone will be able to reproduce your results. This is using ABCD study, which is which everyone has access to. So you can certainly reproduce any of the results that they came up with. And I will be uploading these to a website sometime soon. So these 10 students worked on these projects, and what kind of impact has this had so far? I sent them surveys, self-assessment on rating themselves on their skills and knowledge. I'll give you some results on this. So this spaghetti plots, x-axis time, week 1, and then week 9 out of 10 weeks. They rated themselves on a 5-point scale. Very bottom, 1 is you're not proficient in that thing. 5, very proficient in that thing. And so you have 10 lines here, 10 black lines. That red line is the average. So great thing to see. All the lines went up, except for this person who thought that they were very proficient in art and all of a sudden not as proficient. I do interpret that as I thought I knew everything about coding in R. It turns out that I actually don't. But who knows what it was. Data visualization. These students were learning ggplot and making Shiny apps for their things. And so it's an excellent way, right, for them to really get their hands dirty and producing effective, efficient visualizations and get hands dirty on the data. So this is just some of their results. Here's another one. I never learned about mixed modeling until I was in grad school. ABC data is a multi-site longitudinal study. They had to use mixed models. So certainly everyone got better at that. Obviously, it's an adolescent development program. And the other thing too, right? The first word to our program is equitable. Community engagement was a very important thing in our program. So we had, a lot of things related to community engaged research, and so it was nice to see that that we can have an upward trend on that. So bottom line here is that we can get started earlier. You don't have to wait till grad school. Training can start certainly earlier, and planting the seeds not only on this is how to be a data scientist that produces reproducible methods, reproducible analyses, but also planting the seeds about topics and research questions related to things that is certainly of interest to the people here. So this is my bit about the first bit on training. So I'll go a little bit to Changepoints. Again, this is going to be fairly non-technical. I will also say I want to again thank the organizers. Because when I started working on Changepoints, so it turns out a year and change ago we had a similar workshop in Oaxaca, Mexico, which during that time it led to it created my collaboration with Rebecca Killick, and that's how I got started with Changepoint analyses. And so I'm good, which I'll be showing a little bit of today. We already had seen from previous presentations about connectivity. At the end of the day, you have a bunch of time series, extracted from the brain, you cross-correlate them, you get a correlation matrix. This is an example of that. This came from an American statistician, the American Statistician Journal. I don't know if Shou is here, Shou Chen. Okay, he was working with somebody. This is the figure that they produced, 400 and change regions of interest from the brain, and obtain the correlation matrix and get this. And one thing that's very pronounced and noticeable here is that there's a really nice blocking structure. So essentially, you can think of it as regions of the brain that are spatially nearby or that form, their own kind of network will start to create blocks like this. And so they have this method on how to take advantage of this blocking structure in your correlation matrix for estimating the connectivity. So actually, independent of them, we came up with a method that was very similar. It's similar in the sense that when we were doing lit review, I encountered this paper, and at first I was like, oh my God, they already beat us. They sent us their code, like, oh God, thank God we got the same result. Which is also nice to see. But then we embedded these things into a Changepoint framework. Anyways, regarding this block structure, this is from their data. I used it in my data, and it's not as pronounced, but you can kind of see it. So here, these are regions from the default mode network, left and right broken up, posterior cingulate cortex, anterior cingulate, medial prefrontal, and left and right hemispheres. And those block squares on the diagonal there just denotes these are the parcels from the Glasser atlas within these regions. And so at least within the PCC, you can see that it's bright pink or red. And you can kind of see some left and right symmetry going on. And not so much for ACC and PFC. This is for one individual, by the way. If I were to have removed those block squares, you'd probably only see, say, this and maybe this and this. And maybe you wouldn't have said that there was anything really there. But at any rate, so this is what we want to estimate for connectivity. Dynamic connectivity says, well, these things are changing over time. So this is then when Changepoint comes into play. So if we then embed this modeling framework of estimating block structures and correlation matrix inside a Changepoint analysis model, we would get something like this. So the top left was essentially the same plots before, and then for this particular individual, detected 2 change points leading to 3 segments in time, And then you can kind of see that the correlations got stronger within segment 2 and then died back down segment 3. This will change from individual to individual because everyone's temporal patterns are going to be different. Some people will have no change points, some people will have more change points, and so on and so forth. But from here, what we can then do is extract the number of change points, and then that is now our quote-unquote summary statistic, which we can move to post-hoc analyses. And that's pretty much the gist of the method. So the mixed modeling here is about, we use that in the random effects, then we impose a very specific parametric form to account for that block-like structure. And because we need to be able to estimate these things very, very fast, we use method of moments for this. And then Rebecca has been doing PELT for a decade and change. And we were able to adapt what she did for embedding our block-like structure and embed that within PELT for the change point estimation. So that's a summary of the method. Now I just want to take just the next few minutes on talking about problems. So here's one problem. Can we account for the spatial structure and the data when we are thinking about dynamic connectivity? So what do I mean by that? So in what I was showing earlier with those correlation matrices, each row column in that correlation matrix corresponded to a parcel. They were within big black squares, that's within a region, and then the whole thing was within a network. So what we did, this is results from the HCP-D study. We looked at the number of change points detected across the individuals across these 4 networks. And one of the things you will see here, so the x-axis here, number of change points detected, and then this is just the number of individuals with that many change points. That if we look across the different networks from default mode to reward network and frontal limbic and dorsal cortical striatal, that the number of change points change, meaning that there is some spatial non-stationarity in the data. That different regions of the brain, different networks of the brain will be changing differently between each other. And so Rebecca and I have been thinking about this idea of global change points and local change points globally across the brain, locally within space, and trying to really reflect the fact that different dimensions of your time series will change differently between each other. So this is ongoing work. My PhD student Ellery Island is currently working on this. Another one is HCP-D is a longitudinal study. And I think we had a previous presentation about ADNI, also longitudinal. I've been thinking about longitudinal connectivity for a bit, but it's not really straightforward to me on how do we do this for dynamic connectivity. Can we, obviously if we just extract summary statistics and then go with that, that's fine, but can we do more on trying to do some sort of pooling when we are creating our longitudinal models for change point analyses. This third one, I'm not really sure if it's a statistical thing, but it's definitely an important thing to think about. The impact of motion. People have been thinking about this already for static connectivity, for just good old correlations, and motion is a big deal. Dynamic connectivity, I would argue, is actually worse. And in fact, one of the things that my student Ellery showed was that the number of change points correlated very strongly with the amount of motion of the participant. When she did her HCP-D analysis, we actually restricted to the low motion participants. And by that, I mean those who, whose frame-wise displacement was on average less than 0.3 millimeters. So this is already really, really good, but yet it still correlated with motion. And I've seen that also with hidden Markov models, and certainly also here with change point analyses. But what can we do about that? I have been thinking about, say, well, we could say start censoring the data, but then if we start doing that, we introduce another can of worms. And so those are at least 3 of the many problems I have in mind. And with that, I will end here. So thank you.
We have time for one question.
What's kind of important scientific question you can use in this?
Sure. what kind of questions can you answer with dynamic connectivity analysis? It's pretty much the exact same questions on what can we answer with regular old connectivity, right? And in particular here, we can do a little bit more. It's not just static connectivity, but we can extract more features from it, namely things related to temporal dynamics. So for us, it's not just correlations anymore. At least the feature that we were extracting was number of change points, which is a reflection of the amount of quote unquote non-stationariness of the data, which has, which actually in the studies that we had, we showed that it was pretty good predictor of non-suicidal self-injury and also a good predictor of ADHD.
Hey, Mandy, can you hear us?
Hi.
Hi. Yeah, our next speaker is Mejia from Indiana University, the hidden cost of stringent motion scrubbing.
Okay, do you see the clock or no?
No clock.
No.
Okay, no clock. All right, thank you so much to all the organizers. And I'm sorry I can't be there with you in person, but I'm glad to participate in this great workshop and to see all these amazing talks. I'd like to especially thank Mark for mentioning the importance of thinking about motion when doing these kinds of complex analyses, because that's exactly what I would like to talk about today. And then also to reflect on the questions that Gian has posed to us. So the title of my talk is The Hidden Cost of Stringent Motion Scrubbing. So the alternative to doing no scrubbing or censoring is to do a lot of motion scrubbing or censoring. But I'd like to share with you is that what we have found in some recent analysis is that there are costs to doing that. So to break it down into a few parts, I do hear an echo, by the way. I don't know if there's a way to fix that. We find that more stringent scrubbing actually tends to worsen the accuracy of functional connectivity. In the second part, I'm going to try to reconcile these findings with the more traditional findings of the scrubbing literature, which is that QCFC actually improves with additional scrubbing. And then finally, we'll share some preliminary results on how scrubbing affects BWAS studies. So first of all, if you are not an fMRI person, you may not know what motion scrubbing is. So motion scrubbing is actually a very simple procedure to remove or censor certain fMRI volumes that are thought to contain high noise levels. And this is typically based upon a measure of head motion called frame-wise displacement, which is often combined with the data-driven measured D-bars. And it is often an additional step taken to remove neighboring volumes as well. So preceding or following volumes to the high head motion volumes. So I refer to this as expanded scrubbing. So what's common to do is actually to remove 1 before and 2 after. So you remove 4 volumes for every 1 volume that actually exhibits head motion. Okay, so head motion scrubbing has been around for a long time. I'm not going to go into all these papers, but what I'm showing in color is some of the recent more challenges to the practice of head motion scrubbing. So one of the challenges is that modern denoising methods actually work quite well, and motion scrubbing does not adjust for this. Another challenge is that multi-band data, which is quite common in current acquisitions, requires modifying the motion procedure, the motion scrubbing procedure, and it's a bit difficult to find a solution that is generally applicable. And we also need a lot of data, and we know that motion scrubbing results in throwing away lots of data and can also induce selection bias, which is a problem when we want to study the population. Okay, so to jump into kind of our main findings, we find that more stringent scrubbing actually tends to worsen the accuracy of functional connectivity. And to understand this, we can think about functional connectivity from a simple measurement error perspective. And so when we are estimating functional connectivity, of course, we are only estimating it. So if the true functional connectivity is theta, we observe that with some level of noise. And although it's difficult to come up with a model exactly for how that noise is generated, because there's many factors, we can think of it as being inversely related to the amount of data that went into the estimation. So T is the amount of volumes that we have for this estimation. And the numerator is basically a baseline variance reflecting underlying noise levels in the data. So the thing is that both of these things are reduced by scrubbing. We reduce the noise, but then we also reduce the amount of data that goes into our estimate. So it's not clear whether this error variance is going to increase or decrease just based on this simple model. So we wanted to look at this empirically. So we use data from the HCP retest dataset. That's the Human Connectome project. So we have 42 subjects with nearly 2 hours of resting state fMRI data. So we use a parcellation, a very common parcellation of the cortical surface plus 19 subcortical regions. And the key here is that we established very clean functional connectivity values based on 90 minutes of data per subject with the most stringent scrubbing that anyone would use in practice. And then we produce estimates based on the remaining 30 minutes of data. So we varied the duration from 5 minutes to 30 minutes and held out the rest of the data for the ground truth. And then we quantified the accuracy relative to the ground truth. And then to summarize across subjects, we computed the median absolute error. Okay, so we use common techniques for denoising the data. So we use 2 techniques. One was the 36 parameter regression, which consists of realignment parameters, white matter and CSF, and the global signal, plus their derivatives, squares, and square derivatives. So this is a pretty aggressive form of denoising. And we also use ICA-Fix, which is available within the HCP. And then we scrubbed based on FD with DVARs using the threshold approach of the Nichols paper. And the scrubbing thresholds we considered were no scrubbing, FD over 0.5 millimeters, which is a lenient threshold, FD over 0.2 millimeters, which is stringent, and then this stringent and expanded technique where we do that FD2, but then we also remove the preceding volume as well as 2 after, as well as we also remove any remaining segments with fewer than 5 volumes. So that's an additional step that is commonly taken with this expanded scrubbing approach. Okay, so kind of jumping to the punchline, what we find is that especially this expanded scrubbing approach, we remove not just the volumes where there was head motion, but also any neighboring volumes and the short segments, pretty dramatically worsens functional connectivity accuracy. So these 2 plots are showing for the 2 different denoising methods that we considered, the 36P, which is just based on motion regressors and tissue regressors, or ICA-Fix. In both cases, we find that the FD2 plus, which is the expanded or the expanded stringent scrubbing, really worsens or increases the median absolute error of functional connectivity estimates. So on the x-axis, I'm showing the magnitude of the ground truth. So you can see that the error changes with the magnitude, but overall, the error is just simply much higher with the expanded scrubbing. And then we wanted to kind of understand what were the drivers of this. So we use this equation that I'm showing on the top right to try to get to that baseline variance value that I showed earlier. And so if you look at the baseline variance, it does seem that stringent scrubbing, at least in the 36P case, does reduce the baseline variance. However, we really pay the price when it comes to the sample size. So stringent and especially stringent and expanded scrubbing, this FD2 plus method, just dramatically reduces the amount of data we have available for our estimation. So we're removing more and more subtle noise, and in some cases possibly no noise, but we are dramatically reducing our sample size. So that's what drives the higher estimation error. Okay, and then what happens if we change the scan duration? So what I just showed you is based on 10 minutes. So now we looked at varying scan duration from 5 minutes to 30 minutes. And in order to be able to do a test and assume independent observations, we computed the error at a subject level and then averaged across edges. And so what we're looking at is the percentage change in error versus no scrubbing. And then we performed a paired Wilcoxon test between scrubbing methods and used Bonferroni correction across all of the tests. Okay, so first, just kind of an exploratory plot. We average these median absolute error values across all edges. Actually, average the percent change relative to no scrubbing. And so you can see here is that this expanded scrubbing obviously has just much higher error levels. This is for the 36P denoising. And for the lower scan durations has around 7% worse error on average across the edges versus no scrubbing. And then getting to the test results, these are by duration, to varying from 5 minutes to 30 minutes. And the asterisks show where we have a significant difference. based on these paired Wilcoxon tests. And what we see is that across all scan durations, the expanded stringent motion scrubbing is significantly worse than lenient motion scrubbing in terms of functional connectivity accuracy. So, and even the FD2 method is often worse than the more lenient scrubbing method. So in general, more stringent scrubbing is actually giving us less accurate functional connectivity. And I think it's important to recognize that this data has already been cleaned. We've already used either 36P or ICFX. And so there's not necessarily a lot of noise to be cleaned up after that, the aggressive scrub or cleaning that we've already done. So how can we then reconcile this with the literature around QCFC, which refers to looking at correlations between motion and functional connectivity? So again, QCFC refers to the correlation between head motion, or FD, and functional connectivity. And the premise here is that the presence of motion artifact results in increased correlations between functional connectivity and motion. And so this is computed very simply based on a correlation between the head motion and the Fisher transformed functional connectivity, and it's an edgewise measure. And we can also equivalently compute this through a linear regression slope as long as X and Y have been centered and scaled appropriately. And this facilitates adjusting for confounders. But one limitation of this is that the fact that motion artifact leads to higher FC motion correlations does not imply that any FC motion correlations are attributable to motion artifact. In fact, a 2014 paper identified a motion trait representing true functional connectivity differences between low and high movers. And they found that those differences were not observed within a subject across low and high motion sessions, but really they were attributable to between subject differences. So they refer to this as motion trait that predisposes certain subjects to move more. And it consists mainly of distal connectivity within the with the default mode network. And so it can have something to do with concentration. And therefore, the use of this naive QCFC can encourage the use of methods that might actually eliminate such differences between subjects that are actually real traits. And so what we propose is to instead use something which we call partitioned QCFC to separate QCFC into within and between subject components using repeated measures data. So we, instead of having to fit a mixed effects model at every edge, we instead take a simpler approach modeled after this paper by Guillaume et al. In 2014 for longitudinal analysis, where we split the regressors, the time varying regressors, into within and between subject components. So that allows us to use OLS to isolate the within subject and between subject effects. And so the within subject is what we want to eliminate or at least reduce or mitigate, whereas between subject correlations between motion and functional connectivity could be valid and related to motion trait. So if we look at a matrix of these naive QCFC values, just to illustrate this, if we then use this approach, we can split those values into the between and within subject components. So here you can see that the majority of these correlations are actually being driven by between subject correlations between motion and functional connectivity, and the within subject component is actually quite small. This is based on FD2, which is just stringent but not expanded motion scrubbing. And then if we look at this as a function of scrubbing approach, we see that actually this FD2 plus does not seem to really improve QCFC at all. But especially when we look at the within subject, you can see that that seems to be pretty much mitigated with standard stringent motion scrubbing or even lenient motion scrubbing, which does seem to reduce QCFC within the within subject component relative to no scrubbing. So I think that this suggests that it's not that scrubbing is bad, but that we should take a measured approach to scrubbing versus throwing out the baby with the bathwater. So in particular, expanded scrubbing does not improve either naive or within subject QCFC. Okay, so finally, I want to just briefly touch on BWAS. I'm going to skip ahead, I think, to some of the main takeaways about our BWAS findings. But just to make sure we're all on the same page, BWAS refers to associations between behavioral measures and brain measures, where here brain measures are edgewise functional connectivity values. And it's important to keep in mind both the bias and the variance of BWAS when considering the effect of scrubbing or of any measure that's going to affect the reliability of functional connectivity, because there's both bias and variance that is going to be affected by the reliability of functional connectivity. So previous studies, very high-profile studies, this Mark paper in particular, have pointed out that we need large N to overcome the variance, especially when we have a small correlation value. But also we have this term here, this reliability term, that is also playing an important role on the variance, but perhaps even a stronger role on the bias. And so we looked empirically at the bias of BWAS, and what we found is that there was a substantial amount of BWAS bias being induced because of the poor reliability of functional connectivity. And this is definitely mitigated with longer scans. We have a scan duration on the x-axis, but there's a major effect of denoising technique. So if we potentially are doing too much denoising with the 36P or too much redundant denoising, we are going to pay a price in terms of the reliability of functional connectivity, and then therefore also in terms of the bias on our BWAS correlations. And then scrubbing also has a major impact, or can have a major impact. So in particular, in the 36P case, we find that this expanded scrubbing approach substantially worsens the bias of our BWAS correlations.
Mandy,
yes.
Wrapping up now?
Okay, yes, I'll switch to my last slide. So yeah, so scrubbing has these a lot of consequences for BWAS that I think have not necessarily been top of mind. And so in the last 30 seconds or so, I'd like to reflect on the question that Zhang posed to us, or one of the questions: how can we make statistical methods more impactful in the neuroimaging community or in the imaging community? And what I'd like to ask us to think about is that statistics plays a crucial role at multiple levels. So certainly developing more sophisticated models, which I think many of us excel at, but also in terms of fundamental statistical thinking. And there's been a number of awakenings, especially within the field of fMRI, that have had a dramatic influence on statistical thinking in neuroimaging, such as the dead salmon, Buddha correlations, analytical flexibility, and so on. But many of these have been initiated by non-statisticians. And so what I would love to see is us playing a larger role in some of these fundamental aspects of statistical thinking in neuroimaging. And I believe that we can expand our influence in that way by expanding our scope, by thinking about being involved in these more fundamental conversations, participating in the education and training of neuroimagers, development of software, et cetera. And therefore, I think we will also increase the influence of our wonderful models. Thank you.
Thank you, Mandy. We'll skip to the next speaker and reserve questions from Mandy for later. Our third speaker for this session is Sean Simpson, Wake Forest University. And the title of his talk is Statistical Brain Networks Analysis, Recent Developments and Future Directions.
All right, good afternoon, everyone. Thank you all for coming out. So today I'll just be briefly touching on some recent developments that our group has made for brain network analysis and some potential future directions in the field. So first, I'd like to acknowledge all of the co-authors on relevant work, my collaborators, and the funding mechanism. And so first, I'll talk about the impetus for the development of some of our approaches. I'll then get into descriptive methods briefly for brain networks and talk about network metrics as random variables. I'll then get into three modeling and inferential frameworks that our group has worked on for brain network analysis, and then end with some conclusions. So now, connectivity and network analyses, as we all know, have exploded over about the last 20 years or so and hold a lot of potential in helping us understand normal and abnormal brain function. Now, functional connectivity analysis, as we've heard already today, examines associations between time series and specific regions of the brain, whereas network analysis is slightly different, where it quantifies associations between time series in all regions of the brain to create this interconnected representation of the brain, or a brain network. And the appeal of the network approach is that it allows assessing how systemic properties of our brain are related to behavioral and health outcomes. And while functional connectivity analysis obviously underlies network analysis, the subtle distinction between the two is often overlooked in the literature. For us, the systemic organization of our brains that confer our functional abilities, as we may lose connections, for example, due to some concussion during a sporting event, but compensatory connections may develop in order to maintain organizational consistency and functional performance. And thus, groups may exhibit differences in functional connectivity while still having the same overall network structure. And so here's briefly a schematic for how we generate our networks from fMRI time series. I won't belabor this since I think we're all familiar with this, but we get the activity levels from subdivisions of the brain and look at how these time series are correlated with each other to create our unit of observation, in this case, the network. And this is for the static network case. And so what we'd like to be able to do is relate these networks in some way to endogenous covariates or network features and exogenous covariates of interest, like health and behavioral outcomes. And so in the dynamic or time-varying network construction case, then we just tend to create multiple networks per scanning session to see how the network dynamically changes within a scanning session. And so again, in that case, we'd still like to be able to somehow relate these dynamic networks to endogenous network features and exogenous covariates of interest. And so once we've constructed our network, the next step is often to describe it. And so here we have some example network metrics by category for static networks, and there are a plethora of descriptive metrics for static networks, but here are just some common ones. We have the clustering coefficient for functional segregation and path length for functional integration. We often look at degree or analyze degree distribution for resilience, centrality and information flow metrics, and we often look at community structure. And dynamic descriptive metrics are relatively nascent, but they're developing rapidly. And while there still remains a lot of work to be done in developing network metrics, especially for dynamic and time-varying networks, it's important to remember that we're estimating this network. And so we need to propagate that error from the estimation of the network through to the network metrics for subsequent inference. Because again, these network metrics are functions of an estimated network and are thus estimates themselves. So more formally, if we denote the true network as a set of nodes and edges, and the estimated network is y hat, and we denote the true value of the network metric as g of y, and its observed or calculated value is g of y hat, then we're interested in things like whether or not our estimate is unbiased, what the distribution of our estimate is, what the confidence intervals are for our network metric. And to my knowledge, there have only been a couple of investigations in the literature into this where in both cases they quantified the propagation of error from network estimation to the density metric in particular, which is just a proportion of the number of existing edges relative to the number of possible edges. But this is just one of many metrics commonly used for description. And so this area of network error propagation has barely been tapped, and I believe provides extremely fertile ground for further work for statisticians. I haven't myself worked in this area, but I know that's an important area to work in. Now, when it comes to modeling and inference, once you have your constructed networks, appropriately modeling and drawing inference from network phenotype relationships requires the integration of statistical tools with network neuroscience methods, which I believe presents us with a unique challenge. And while many approaches have been developed, few integrate these two domains while also allowing for group and individual level assessments. And additionally, the optimal methods are going to vary with the rapidly evolving questions of interest, and this necessitates a suite of alternative complementary approaches. And so we've attempted to address this need somewhat by developing three complementary statistical frameworks: a mixed modeling framework, a distance regression framework, and a hidden semi-Markov modeling framework, but a lot more work is needed. And in subsequent slides, I'll sort of give a high-level overview of some of these methods without the details. And again, in general, or one of our aims anyway, is to try to assess the effects of multiple variables and network features on the overall network structure. So if we denote our network of subject I at time point t during task L as YITL, we'd like, one aim would be to try to model the probability density function of the networks given our covariate information. So one such approach we've taken to attempt to doing this is we worked on a two-part mixed modeling framework where we modeled the probability of an edge existing as a function of network metrics and covariates and the strength of that edge if it exists in a similar fashion. And so in statistical notation, we just use a version of a logistic mixed model for the presence portion and a Fisher Z transformation on the strengths and modeled that in a similar way. And then these theta parameters contain subject-specific and dyad-specific, where a dyad is just a pair of brain regions, a random effect and random error terms to try to capture some of the dependence in these networks. And so we then extended this approach from the single network case to the multitask network case. And here just L denotes task. And then we also extended it to the dynamic or time-varying network case, where we just introduced orthonormal polynomial terms in the fixed and random effects to try to capture the dynamic trends in the data. And so as an example, in this last development, we use it to analyze differences or dynamic changes in those with higher intelligence scores and lower IQ scores in the HCP young adult data. And briefly what we found is that there were dynamic differences in modularity, and basically those with higher IQ scores, their dynamic changes in modularity tended to stem more from the strengthening of within community edges, and those with the lower IQ scores, their modularity changes tended to stem more from a weakening of between community edges. And so this sort of architecture here definitely allows for better information flow. And so for future work within this domain, we'd like to try to integrate our multitask and dynamic models so we can look at dynamic differences between groups across tasks. And so the next framework I mentioned is a distance regression framework, and that was basically motivated by our desire to assess spatial, topological, and distributional consistency of key node sets across groups and individuals. So for instance, in this graphic here, we're looking at hub nodes, or the top 20% most connected nodes. And we can tell that it looks like their location is different in the two groups by these overlap maps, but we wanted to be able to formally assess this. And so recently we developed this framework that allows doing this while controlling for confounding variables. So here we're looking at the distance between networks of I and J. So if we're looking at overlap, for example, we could use the Jacquard as our distance metric. And we're looking at it as a function of confounders and covariates of interest. And so, for example, if we wanted to test whether or not the spatial consistency of hub nodes, it varies by IQ after accounting for age, sex, and treatment group, we'd use the Jacquard metric as our distance and assess whether or not beta 2 is equal to 0, be our null hypothesis in that case. And so in this formulation, the errors become the problem because they render the standard F test inappropriate. Because again, remember, we're expanding our N subjects of data into N choose 2 pairwise comparisons. And so we have correlated data. And so within this framework, we assess several estimation and inferential approaches. And we found one that worked really well and beat out the main competitor in this context, which is MDMR, in terms of power and type 1 error control. But I'll leave that out here for the sake of brevity. And then we also extended this approach to deal with the HCP test retest data that Mandy mentioned in the previous talk. And so basically here we are looking at the distance between the networks of subjects A and D for task B over all repetitions C and F. And again, this is sort of the setup for that test retest data that motivated that Mandy was talking about, where each subject was scanned during these 3 tasks in 4 repetitions. And so what we proposed for that setup was what we call the 3 Bantor, which is a mixed model for multitask brain network regression. And basically here in this setup, the ID variables are indicator variables for comparisons between distinct individuals A and D on the same task B with this corresponding random effect. And we found that this approach outperformed MDMR and outperformed other approaches we had developed. And so in future work, we're looking to extend this framework to be able to account for familial correlation. This is particularly motivated by the familial structure in the HCP data. And now work by my colleague, Heather Schoppel, who'll be actually talking about this in the next session. She's looking at hidden semi-Markov modeling for state-based dynamic brain network analysis. And so here, here's like a graphic illustration of that sort of model where we assume that the unobserved states in our data evolve according to a semi-Markov process characterized by state transition probabilities, the red arrows, and sojourn distributions, the dashed lines, or how long you spend in a state. And so here's basically how the model is formulated. And so basically each network state has its own mean activation and its own covariance matrix among the ROIs. And the complete data log likelihood is used to obtain group level estimates of network structure, transition probabilities, time in each network state, as well as to conduct inference about these estimates. And so as an example, here we have on the left estimated sojourn distributions of kids with ADHD and typically developing kids for 2 states that reveal statistically significant group differences. And on the right, we have correlation matrices for each of these 2 network states. And so Heather will be talking more in the next session about potential avenues for future research within this framework. So just to close out, so network analyses may revolutionize our understanding of brain function, but the methods need to keep pace with the rapidity of scientific inquiry in the field. And given the variety of scientific questions of interest, a multifaceted suite of complementary approaches is important to continue moving the field forward. And so we've tried to add to this suite. And in the spirit of this workshop, if any collaborators are interested in these methods or relevant or related methods, just reach out to us. There's a software package for the mixed modeling methods, and there's GitHub code for the distance regression methods. And again, a lot more attention needs to be paid to error propagation from network estimation. And thank you for your time.
We have time for one quick question for Sean.
For the distance regression, you imagine distance between networks.
Right.
What distance?
So we actually looked at several distances to, so the Jakar distance when we were interested in overlap, for instance, between 2 networks. We looked at Euclidean distance. Actually, Euclidean distance actually had the best properties overall in terms of type 1 and type 2 error. But we looked at some Minkowski distances. We looked at a variety of distances within that framework. So it's not limited to one distance. It more depends, I guess, on your question of interest.
Oh, okay. Topology distance may improve the performance.
Okay, that's good. I'll look into that. Thank you.
Thank you, Sean. Our final speaker for the session is Tingting Zhang from Pittsburgh. Her talk is on the analysis of functional brain network changes from childhood to old age.
So first I want to express sincere appreciation to Jen and also Home2 for their great efforts in organizing this workshop so that it can bring us together in Chicago. It's great to see many friends here and also meet new researchers, young researchers here. So I'm going to talk about the study of functional brain network changes from childhood to old age. So Jen sent us an email, asked us to address 3 questions. So the first question is, what are the most important problems we should focus on in next 5 years? Okay, since I'm giving only 15 minutes, so I just talk about the one. I think the aging research is definitely will be a very important areas for all the human lives. So first that everybody will be affected by aging, unfortunately, and actually all the developed countries and many developing countries are facing the big challenges of aging societies, and we all will age, and eventually, unfortunately, so in recent years, the aging research has evolved its focus from merely extending lifespan to promoting healthy aging. However, the growing prevalence of age-related brain diseases, especially among the very elderly, create challenges to achieve healthy aging. So you can see in this field, so aging research, not everybody cares. So you will definitely have a great impact on the entire human societies. Also, this field generated a lot of neuroimaging data. So statisticians working on neuroimaging can find a lot of data resources and collaborations in this field. So coming back to my own research on the aging. So when I first got into this field, my question was, okay, if I want to achieve healthy aging, what my functional brain network should evolve with age? So what are the normal changes in the functional brain networks? To address these issues, we analyzed resting state fMRI data aggregated from 3 human connectome projects, including HCP young adults, HCP development, and HCP aging. So in total, we analyzed around 2,300 subjects, age from 8 to 100 years old. And so this age, so we want to evaluate how the whole brain functional connectivity changes from age 8 to 100 years old. So in the literature, several methods have been developed to address these problems. Because of time, I just talk about the most relevant one, independent regression analysis in adult actual connection-wise analysis. So the approach is very simple. So for every pairs of brain region I and J, we will measure the functional connectivity between them in every subject. Then we use it as a response variables and regress it versus subject covariates such as age and sex, and maybe also other covariates. So this method is highly flexible, so you can evaluate the functional connectivity changes for all pairs of brain regions, for all connections. But the problem is that the brain has hundreds of regions. Using this method produces enormous amounts of FC trajectories. So for human brain, it's very difficult to tell, okay, which brain regions demonstrate distinct patterns of age-related functional connectivity changes, and which regions actually age together in terms of the functional connectivity. So to address this issue, we propose this so-called cluster-enabled regression analysis. So we introduce cluster labeling for each region. So based on this model, we try to identify the regions that show synchronized substantial changes in functional connectivities. So based on this method, we no longer need to evaluate FC changes for all pairs of brain regions. We just need to look at the FC changes between these identified region clusters. So here is the because of time, I skip all the technical details. Just talk about the real data analysis results. So we found several interesting things. So first, regions that show synchronized substantial changes in functional connectivity are generally from the same functional networks. So here we show this. So we show the identified region clusters for different functional networks. So region in the same cluster, in the same color, are identified to be in the same region clusters. So you can see that, okay, so those large, so first is that regions shows synchronized substantial changes in functional connectivity generally from the same functional network. However, large functional networks like single operator network default mode, so their region can demonstrate different age-related functional connectivity changes. So this result shows that we cannot take regions in the same functional network as a whole. We have to treat them differently. And because of this result, we classify our FC trajectory into two types: within network FC trajectory and between network FC trajectories. We find that within network FC changes in a very similar way. They generally monotonically decrease with age. So to illustrate those functional connectivity trajectories, so in each of this plot, we use one region cluster as a seed cluster and plotted in black. So for example, so cluster A in visual network is treated as a seed cluster here. And if the functional connectivity between this seed cluster with the second cluster shows a substantial changes with the age, so we plotted associated FC trajectory in the same color as the second cluster. So for example, here this one, so FC between cluster A and cluster B have monotonic decrease with age and very substantial changes. We plot the associated FC trajectory in in the same cluster as a cluster B. And here, this solid line indicates the population mean FC trajectories for males. And it's actually 95 confidence intervals for population mean FC trajectory for males. And the dotted lines are for the females. So the blue and the red dots are the FC measurements for individual subjects. So you can see that so within network of functional connectivity, generally monotonic decrease with age. This is pretty consistent. However, between network of functional connectivity shows diverse patterns. And generally, so for the networks including single opercle network, sensory networks like visual, so motor, auditory, and including language and dorsal. So FC between these networks, their trajectory tend to be increasing, or sometimes they have a slight decrease after age 70 or 60s. So they have inverted U shape. So inverted U shape and increasing pattern are very common. However, we also observe this decreasing between network FC trajectories. It turns out that this is a very unique one because this is a FC between two spatially neighbor ranges, such as this seed cluster in black and this pink motor cluster B. So we find that the FC between spatially neighboring regions tend to monotonically decrease with age. And the default mode network has very different FC trajectories. So we only find that for substantial changes, we only find increasing patterns between default mode and a single opercle network. And the connection between default mode network with other network generally monotonically decrease. So in summary, between network of FC, age-related changes in between network FC have diverse patterns, and the pattern not only region and network specific, but also depends on the spatial distance between regions. And in addition, we also find clear differences between female and male brains. So typically, females have a higher FC than males within the default mode networks. However, males have a higher FC across many different networks. So this includes the connections between default mode and a single opercle network. These are very few FC connections. You can clearly see the population mean differences even in childhood. So generally, the female and the male's functional connectivity are similar at childhood, but the differences increases as they age. So, and we also find that males have a higher FC than females within the somotor-motor network connections, and also within the auditory connections and the connections between the somotor-motor with single opercle network and the somotor-motor with visual. So in other words, males, especially elderly males, have a higher FC across more networks. We think these results correlate with the observation that females tend to be more vulnerable to Alzheimer's diseases, possibly because females tend to mainly rely on within default mode network to maintain cognitive function. And unfortunately, Alzheimer's diseases tend to target default mode networks. So because of time, let me summarize this result. So in this work, we identify regions that show synchronized substantial changes with age. So basically, we find the regions have age together in terms of functional connectivity. We also show different patterns of FC age-related functional connectivity changes, and we also observe differences between especially elderly female and elderly male's brain functional networks. Okay, I want to thank my collaborators, Professor Alfonso Silva, Stacy, and Dana from University of Pittsburgh, and also Greg Carter from Jackson Laboratories, and my two students, Yao Tian is actually my previous student who is currently a postdoc with Professor Ying Guo and Emory University, and my current PhD student, So Dan, who did all the work for me. So they did a lot of pre-processing. So we're talking about the data pre-processing. So this is related to the second question, how the statistical community improved the data pre-processing and data sharing. I think data pre-processing and data sharing is very important, especially for small labs. So for small labs like me, like my lab, I have very limited hands. So even for pre-processing this 3 human connectome project take us a lot of times. I can imagine that similar problem affect other small labs. So if we can improve this data sharing and data pre-processing, we can speed up the research development, especially for these small labs. So the question is how the statistical community improve it. I think if we want to improve it, first we need to value it. We need to appreciate. In order to appreciate statistical data pre-processing, and we need to promote data-intensive research. You only know its importance while you're doing it. And because of time, I want to go into that details and why we didn't value it. But I'll talk. So the last one is that how we can make the statistical methods more impactful. I think the simple answer is doing interesting problems, doing the problems the scientists outside the statistical care. So if we can solve the problems that they couldn't solve, or even deep learning couldn't solve, they eventually would value the statistical methods. So taking my collaborators as examples, so and Dana is a biostatistician. For others, they are actually neuroscientists. They haven't had experiences working with statisticians. But in the beginning, so you need to reach out and tell them what are the right statistical methods and analyze the data for them. So when they see that, you can get interesting results. And my collaborators say, oh, I finally understand what a statistic is doing. Even though I don't think that he really understands it, but at least he sees the usefulness of the statistical method. So reach out, communicating, and also I think it's very important that we can define scientific questions. So most of the time, at least for my experience, my collaborators don't know what's the right statistical question for statisticians. So if we formulate scientific questions and they find that, oh, it's very interesting and we can use statistical method to address those questions, they will eventually value the statistical methods. And thank you. We have stopped here.
Thank you. Questions for Tingting before we open up? Yes, please.
Just a question to do with interpretation. Why do you think women display increased activity in the DMN network compared to men? Do you have any smart guesses there?
So the fundamental is mainly related to hormone. So some explanation because there's a related hormone will cause the differences and default mode networks. That's one reason. And also it's possibly related to, I don't know, I don't have an answer for that. People find those differences. I think and I also find the differences gets larger as people age. So clearly there are some environmental factors affect it. Yeah.
So you get the the difference between male and female in terms of
Population mean.
I mean the function connectivity, but based on the the HCP data, have you validated on AD since you have some observation on the relevance to AD?
Yeah, we did that. I didn't have time to show this. So we are not able to achieve the, just based on the FC, we are not able to achieve at the individual level. But the population mean level, we definitely say that in the AD data, females have more abnormal connections than males. It's very obvious. Even for mild cognitive decline subjects, you also see that. Yeah, we use this as a prediction intervals and to compare with AD. But the FC unknown is not enough to distinguish AD subjects, and it's just at the population mean level.
Great talk. Yeah. So you put together, aggregated, concatenated like 3 different data sets, right? And so you sort of see a banding in your data set, like from the young adults to the, and so it's a little, I found it a little surprising that actually the young adults seem to be much noisier than the older, right? Isn't the variability in that first band that's more dense, around 20 to 40?
I think it's because more dense, because they only from 8 to 21. So that's 600 subjects. But from 35 to 100, you have 600 subjects. So
Yeah, I know why it's more dense, but doesn't it look like the variability in the young adults is higher, or is that an illusion? It looks like there's a higher variability there, which I would find surprising, because I would think that there would be more variability the older you get, and that doesn't seem to be the
but
case.
young children tend to move more.
Well, 20 to 40-year-old aren't young children, right? Well, maybe when I was, but
You mean this here or this here?
yeah, that band, yeah, it doesn't look. I mean, it might be an illusion, but it just looks like it has a higher variability. Yeah,
I'm
maybe
not
I'm
so
wrong.
sure. I think it depends on the connections. Is that the case?
So you're wondering about the variability of
Well, it just looks like
connections or the
it's a
band?
wider band, like it's higher variability, right, in the y-axis.
I think it's an
people pass
illusion at the
away at the
numbers.
end.
Yeah, that could be true, but still, I would, yeah. Curious.
More sample size came with more outliers.
Yeah,
Perhaps, yeah.
Thank you, Martin. Questions for either Tingting or the other speakers? Sean, Amanda, and Mark.
Oh, man.
And the other guy.
Well, I have Yarek.
So Fernando, I have a question to you, Sean. So you were looking at the global measures of the networks, yes? Have you also explored something, more specific to either subnetworks or, to regions that you might be of interest?
Yeah, so for the first framework, the mixed modeling framework, we've also looked at sub-network analyses. So we had, an interest in specific sub-networks, so we incorporated covariates to allow assessing that. In the other frameworks, we haven't done that, but in that first set, we looked at sub-network differences.
I have a question for the speakers on functional connectomes. It's been a while since I actually handled fMRI data. I seem to be missing the phrase effective connectivity. So what happened to that? Is that not okay? So that's not
Solved.
It's not reproducible. That's my feel for it. You fit a vector autoregressive model, those coefficients are not reproducible.
it's still interpretable.
I mean,
If
you
it
can
was
interpret however
reproducible,
way you
it's
want.
interpretable.
Well, I think it was Carl Friston who pushed it as like there's effective and then there's functional. And
Yes.
effective is really capital C causality, and I assume that most people kind of just back off from that because we don't really believe that. And so once you take the capital C causality, it doesn't really matter. You don't need to distinguish them. We all know it's all functional correlativity. It's just correlations. We do the best we can with that.
But there's still something about activity in one region at a previous time point. You cannot predict activity in another at a future time point. That would still be a. Isn't that still important in hemodynamic responses?
I mean, personally, I think it is, especially when you think about the autocorrelation in the data. I mean, I'm sure that there's definitely lag correlations going on. So I think it's still important, but I think that the use of the term has kind of faded.
I think it's important too, and I guess there's an issue of whether or not we have the models for it, but also do we have the data for it? Because a lot of people say we just don't have the temporal resolution to pick that up.
Can I add that I think that you have to assume that the hemodynamic response is the same across the brain, which we know isn't true. I mean, or you feel you have a model for it that can capture HRF differences. And I'm not as up to date on the effective connectivity models, like the dynamic causal modeling, but that always seemed a bit hand wavy because it seems like we don't really know what the delay is in different areas of the brain.
Thanks, Mandy. We have more time. Any more questions? Derek?
Since I started one, I'll have another
Might
one.
as well.
This is for everybody as the speakers, but also relating to the previous talks where there was a question at what stage of a career should you address certain problems, and I forget now who asked the question, and this was one of the earlier talks, but also Tingting, you mentioned the small lab versus large lab. So for me the question is what problems should be addressed by bigger labs versus smaller labs, because as you were saying with processing of HCP data or if we go even further with like UK Biobank, you need a lab, and maybe Hong too, maybe you can do it, but you know, not many of us can do it within, you know, smallish labs.
I don't agree with that.
Okay.
You can do it, or
I think you left in the smallest lab.
If the curve is a cup, then you should do it earlier.
No, so this is the stage of the career, but you know, the size of the lab, because this is to Tingting's point, you know, that it would be great to have this data processed so others can use it. But again, if you don't get credit for that, you know, who will do something for free? It's not only money, but it's also time.
Is to partner with a group that's already done that, right? So I've seen that Jan Kang in Michigan, so he's partnered with a psychiatry group that's done a lot of the pre-processing. So I think that's a good way to get started.
I can share my personal experience and opinion about this. And I think at an earlier stage of our career, we usually don't do it, right? We just do statistics. And after several years, I find that I strongly depend on my imaging collaborators, okay? And I force myself to do it. Then we just gradually get familiar with all the pipelines and build this up. You don't need too many people at the beginning, one or two, okay? And just make sure you understand everything, then build it up. Well, if you know how to process one data set, then you can do 100 in a year. Then you gradually do 1,000 and 10,000. The most important thing is your CPU is free or not. Now UNC is the CPU is free. I don't need to pay money to do that. We don't need to use GPUs. That's the reason we think for all this data preparation on pipelines, we set up all the pipelines. And also you can collaborate with other people, that's of course. But for me, I set up everything within the lab, and then we'll process all the data by ourselves. Even for UK Biobank, it takes a year to process it. It depends on different specific pipelines. Sometimes it's time consuming. But these days it's not that much because they have the foundation models for the segmentation and ray tracing. That's usually the segmentation much faster than before. Also, the ray tracing the same thing. And then it's not about. I think it's not about size of the lab. It's when you started to work on it, it pushes you forward. Just do it, okay, by yourself. I think because it's very important. I don't depend on other lab to wait for them to process data for me. You know, I just ran a data analysis and waited for 1 or 2 years. Now the paper already published it. Our group already finished everything. That's what I did in my career. I just forced myself to do it. I think it's a great value for you, for anyone. It's not size of the lab.
Can I ask a follow-up question?
Go ahead, Wendy.
So I'm just thinking about the role of the statistician versus the role of the image processor. So it almost feels like a bit of a one-way street that statisticians are often asked to contribute our expertise to practice, right, to applications. But we also, it seems like we also need the expertise. We need image processing expertise if we want to really have our hands in this field in a meaningful way. I'm wondering if anybody has any success writing into grants some funding for a student in an image processing lab or some kind of research training image processing lab that could assist with some of that work.
I think in general, I think for statisticians, I don't distinguish us from these people are doing the imaging analysis, because all the image reconstruction, image ray tracing, image segmentation, essentially is statistics. If you think that way, right? You know, I do some of the DTI ray tracing or stuff with Mejia and also Zhengwu, right? We developed the pipelines.
I think the problem was that a lot of times people divided like this is statisticians and this is not statisticians. I think those lines are blurred. We have to do what we have to do to solve the problem.
Yeah, this is all this data analysis. If you think of data statistics about data analysis, everything is about. You think of that way.
Dana?
I completely agree.
Oh, sorry. Dana has a.
I was gonna
get to Mandy's question, in terms of writing statisticians into the grants or graduate students, I do that all the time because when you collaborate with large imaging labs, so you give them a little bit of, part of the grant. So you write them in as your collaborators, and then we do write students that are part of the, for example, PET imaging centers, but they work with me and they're in the different departments. So that is perfectly fine. I think that is the best way to kind of get into the question, how do we contribute more with the methods to the actual field? And then the other thing is, Hong too, when all these little labs, when they process their data, what I've seen is that the results are not generally the same. And we've done that with large studies. We process the data the same way at Pittsburgh, and we process it the same way at Mayo, and we still see 40% variability in the results. And I think it has a lot to do with just very little tiny things that we do when we run those algorithms. And I'm sure Mandy has seen that, and everybody else that is in this room.
That's the reason I need to do everything by myself. Because I quite good control myself.
Well, the one scary thing, and I'll mention it in my talk, is that at Oxford, they have this pipeline that they put all the UK biobank data through, and then they're asked to make a containerized version of the same. So same person building the same pipeline, but instead of just being on a cluster, inside a container that can be shipped off to a cloud, and the results are different.
That's like that classic freezer, for example, when they
Exactly.
upgraded the operating system and all the results changed.
So everybody, our session is officially ended. You can of course keep chatting at the coffee shop, coffee area. But thank you all. Thanks, Mandy, for joining us.